And this screencast is going to show you how to use the UCSC Genome browser so that you can visualize epigenomic data, explore publicly available data as a part of the ENCODE project, and at the end, make a somewhat publication-ready figure from the result. So the point of this exercise is going to be about visualizing an enhancer in the human genome. You'll want to introduce the concept of enhancer during class, but just to reiterate, it's a non-coding unit of regulatory DNA that can fold over large genomic distances, many megabases, and in three-dimensional space fold over to affect gene expression at another locus. In this case, we're going to be talking about the MYC enhancer. So MYC is a very potent oncogene, and SNPs have been found in the human genome that affect enhancers of MYC such that they have people that have this SNP have a higher increased prevalence of cancer. And this is shown pretty definitively recently in a science paper, and I'll show you a figure from this right now where this enhancer was deleted in mice, and the mice that are wild type for this enhancer develop colon polyps like this, and losing just this one enhancer is protective against cancer. You can see that quantified here. And so the point of this exercise is going to be to go through the UCSC Genome browser and visualize the MYC gene locus and then the enhancer that's over 300 kb away. So what we're going to start with is going to the UCSC Genome browser. And you can get there by going to www.genome.ucsc.edu and clicking on the Genome browser and entering MYC. And that's going to take you to this screen, which admittedly has a lot of information on it. And what we want to do is eventually end up with something that is much more simplified and much more kind of schematized and looks something kind of like this, where we have the MYC gene locus right here, we have the enhancer chromatin marks that are associated with it, and we're going to talk more about this as we go, and then just depict that it folds all the way over to touch and express gene expression of MYC. But we'll save that for later. For now, we need to clean up these tracks. So there's a brief explanation of the UCSC Genome Browser tracks in the assignment, but basically what you're seeing is a bunch of different columns kind of layered on top of each other here that you can select individually to alter the depiction of the genome. So the first thing that we're going to do is just start deleting things off of here. So you can right click, you can start hiding tracks, and we're going to be hiding things left and right at the beginning and then activating stuff that we need. And what we're going to leave is just a depiction of the genes, in this case from RefSeq. And that's all we're going to have for a start. Now we're looking at the MYC gene body right now, and we're actually going to change. Each track has the ability to be shown at different configurable levels of display. So we want to put the gene track here under pack, so that we can see the direction of transcription of the MYC gene right here, and you can see the scale. We're looking at maybe 5 kb here, and we just want to keep zooming out and zooming out and zooming out, because the MYC enhancer is about 350 kb away from MYC itself, and it's located right around here. So what you can do when you have a very large amount of the genome that is selected is to drag individual parts and it will zoom in. So I talked about the enhancer, and what we want to show is the marks of this enhancer. And I'm not going to get too far into the chromatin marks that are indicative of an enhancer, but what I do want to say is that those marks have been experimentally determined as part of the ENCODE project, and they're available to be activated on this track. So this relatively simple track that we have right here with just the genes, we're going to fill that in with other information. And what you're seeing here are different tabs for all the different tracks that you can activate. And what I'm going to encourage you to do is click on many of these and just explore for yourself to see what kind of data is available to you to look at and to integrate with your own data potentially. But for us, I'm going to minimize a lot of this because we're going to focus on gene regulation here for the most part. And what we're going to do is go to the ENCODE histone modification tracks. And we're going to go in, and the Broad has done a bunch of ChIP-seq experiments. And you can see that there is this table here depicting everything that you can activate with a clickable radio button. And on the side is a bunch of different cell types. And on the top here are a bunch of different chromatin marks and different DNA binding proteins and chromatin modifiers that you can activate. For us, we want to see what's going on in an epithelial tissue like the prostate. However, there are no available prostate cell lines on this track, so what we're going to do is take what I would consider the next best thing that's available, normal human epidermal keratinocytes. Any of these things, by the way, is clickable, and it will give you more information about the cell type itself. There are documents as to how these cells were cultured and how they were fixed for these different assays. You can read all about that. This is really a wealth of information. And what we're going to look at is H3K4 monomethylation, H3K4 trimethylation, and H3K27 acetylation. Again, I'm not going to get too far into it, but just take my word that having an enrichment for these two marks right here and a depletion of this mark is indicative of an enhancer. So then we'll go back, we'll submit this, and now you see a bunch of additional tracks that have been added in. So the first thing that I want to do is change the visibility of these tracks, and you can just again right click right here and configure these such that we can display them to be full instead of densely full instead of densely represented. And I'm just changing a few things here as my computer chugs along to record this. Okay, so what you're seeing now is ChIP-seq tracks for H3K4 monomethylation, trimethylation, and H3K27 acetyl. You're seeing the actual ChIP-seq reads displayed here in wiggle format, and then you're seeing in bed format, which is just chromosome start-stop, you're seeing the peaks that are called for these represented in black and gray, in grayscale. So what you're seeing here at MIC is you're seeing H3K27 acetylation peak, H3K4 trimethylation peak, right along the whole area of the gene body. The enhancer that we're talking about is right here. Now the reason that I know that is that you can activate another track that is available here in phenotype and disease associations. And this is a very useful track, which is the Genome-wide Association Study Catalog. And I'm going to activate this, and what it's going to do is it's going to show in little green bars here SNPs that have been shown to be associated with a given disease or cellular state. So I happen to know that this right here is a SNP that predisposes people to prostate cancer. So what I want to do is go from this now to something like this. And there's a few things that we can do to generate this type of figure from something like this. But first, instead of having all of these different ticks that are there, I want to just be able to upload our own custom track to be able to keep track of just the things that we're interested in. And so to do that, all you need to upload your own data to the UCSC Genome browser is to have a file like this, which is chromosome, start, stop, and then some sort of name for this particular interval. And this fourth tab delimited column here is optional. So what we can do is use this file. I'm going to select this, and we're going to say add custom tracks. In this case, we can just paste in this data, but oftentimes if you have thousands or hundreds of thousands of different intervals across the genome that you want to represent, you would upload a file that's in this format. We're going to submit it and then rename it so that we can recognize it on the Genome browser as our own. We'll go back to the Genome browser, and by default it has zoomed us into this exact SNP. You can see a single base pair here is represented, and we're very, very far zoomed in. So what I'm going to do is go back to Nick, and then we're going to zoom out. And this is a common thing that you'll do. You'll be kind of playing with different levels of zoom to try and find what you're looking for. But that's the beauty of being able to upload your own data. Is we can see that this is really, these are the only two data points that are on our track. And we just want to visualize those. So again, we'll zoom over this. And now we have a bunch of our data. And by default, when we selected Nick, it activated a track that we don't want anymore, which is the UCSC genes as opposed to the RefSeq genes, so we'll get rid of those. We'll get rid of the GWAS catalog. And now we have this. So at this point, we're ready to export this because we have all the data tracks that we need, and really now it's just about making it, quote, look better. So there's a few things that we can do. Number one, these light blue vertical running bars, we don't want those. So we can go to configure right here. We can take those away. It's also a little bit distracting to have descriptions above each track, so we'll take those away. And then as we have it right now, it's a very long figure that's difficult to put in a manuscript, as many of you will find out. So we'll change this to something that's more manageable, to be about 600 pixels. Change the text size, because most journals want you to have at least text size of 10. And now we've got something that is a little bit more manageable. So at this point, we are going to export this and be able to work with it potentially in another software. So we go up to View and select PDF or PostScript file. And there's two ways you can do it. You can directly export a PDF, and for the purposes of this assignment, that is all that you are required to do is just export a PDF of your locus with your data that you choose. Or you can get this as an EPS file, and you can bring it into something like Photoshop or Illustrator, and you can build in different things. You can change the color of the text. You can delete certain things that aren't relevant to you anymore. You can move them around, and you can add in things on top here. So at this point, you can download your graphic and save it to your computer, and this is what you will turn in once you've activated your stuff. So have fun exploring the UCSC Genome browser, And please remember, if you have questions, to post them in the comments so everyone can benefit and see the question and hopefully the answer. So thanks a lot, and have fun.